Hi there, guys. I wanted to do a quick screencast to review the study guide for the final exam. So your final, again, is going to be on, if you're in person, your final is Wednesday, so tomorrow in class. If you're distance, your final is going to be on Thursday in Zoom. So on Friday, we went over natural selection. Go ahead and cross out 25. We're not going to be talking, or you're not going to be asked about Darwin's postulates, but definitely natural selection. Vocab heavy, so understanding the difference between genotype and phenotype, fitness, what is selective advantage, and there will be some genetics questions as well. So this was Friday. Going up, Friday we finished with 23. So we went over the Punnett square, set up our cross, and we have our possible alleles in the outcome. Our possible alleles in the offspring. So our genotypes versus phenotypes, what they look like. Genotypes is going to be what's in the DNA, so those alleles, either dominant or recessive. We also are going to work out number 24. So I've highlighted and underlined key information that you guys need to pick out of these problems when you are solving them. So light blue skin is the dominant trait for body color. And it says that he is homozygous, meaning he has two of the same alleles. If he's homozygous and he's blue and that's the dominant, then we know that his genotype is going to be capital B, capital B. So the problem continues. He married a nice girl with light green skin. So we underlined that trait. That's recessive. And I set up a key here for capital B to represent dominant and lowercase b for recessive. So once we figure out our cross, if we know that she has green skin and green's recessive, we know that lowercase b is her genotype. So this is our cross that we put into our Punnett square. And we cross these, and these two genotypes, the genotypes of those two parents, are going to have a heterozygous genotype for every occurrence of reproduction. So we see that we end up with capital B, lowercase b. That's the genotype of the children. And phenotype we know is what is expressed. So capital B, lowercase b, a heterozygous genotype, is going to mean that all of their children are blue because it's the dominant allele. If there's at least one dominant allele in a genotype, it's going to express that trait. If there's two recessive alleles, that means it's going to express the recessive trait. All right, so they have a 100% chance of having a child with light blue skin and a 0% chance, no way, could these two genotypes result in a child with green skin. And again, we went over that on Friday in Zoom. Okay, so going backwards. So we're going from the most recent content to the oldest content. So again, just understanding the difference between gene and allele. So the gene is going to be the segment of DNA that codes for a specific trait. For example, there's a gene for fur color or a gene for eye color. So the allele is the different variations of a gene. So in our mice example, there was the variation of dark fur or the variation of light fur. Genotype, again, is the genes or the DNA. So that's what's in the nucleus of every cell of our body is our DNA. We can't see that. We use letters to represent genotypes. For example, capital B, capital B, capital B, lowercase b, and two lowercase b's. We would say homozygous dominant, heterozygous, or homozygous recessive. Phenotypes, the physical trait that is expressed that we can see. Homozygous, that means the genotype has both of the same alleles. So an example would be homozygous dominant or homozygous recessive. Heterozygous is different. You have one of each. So you have one dominant and one recessive. Quickly to go over crossing over, I found a nice image here. Crossing over is when your homologous chromosomes exchange pieces, and finally you have a recombinant chromatid here. So we see that one section from the blue chromosome is now on the pink and vice versa. That results in variation. That is why in meiosis you end up with 4 genetically different haploid gametes. So we call this gene swapping. It contributes to genetic variety in sexual reproduction. Major differences between mitosis and meiosis, I would look for a simple diagram. So basically, mitosis is going to result in 2 diploid identical body cells, and meiosis is a type of cell division used in sexual reproduction to create the gametes of sperm and egg. So meiosis makes 4 different haploid gametes, meaning half of the chromosomal number. So they can combine in sexual reproduction to make a diploid cell. 12 and 14 are related. I'll start with 12. So here's an image, a diagram representing the process of translation, also known as protein synthesis. So our mRNAs are single strand. We have our ribosome as the gray molecule that the mRNA is going through. tRNAs are right here, bringing in those amino acids. So number 14, messenger RNA is a single strand of nucleotides. So nucleotides are represented here, used for protein synthesis. It's formed in the nucleus, leaves and travels to the ribosome, carrying the instructions for the amino acid sequence. Just recall that RNA and DNA are made up of nucleotides. So here's an image of a nucleotide. We have our nitrogen base is usually represented, it could be on either side, usually like a rectangle shape. Those nitrogen bases are those A's and G's and T's and C's in base pairing, or it could be U for RNA. Your 5 carbon sugars in the center, it's that hexagonal shape, and phosphate's always your circle. Sorry guys, that's kind of going crazy. All right, so we here have some just basic definitions. DNA replication is when DNA makes a copy of itself. It happens in the nucleus. Remember that A and T and G and C go together. Transcription is when RNA is made from DNA. That also happens in the nucleus because DNA cannot leave the nucleus. RNA is 1 strand, and remember A goes to U and C goes to G, so there are no T's in RNA. Let's go over a couple examples of how to use our codon wheel. So let's see here. So remember that the genetic code is read 3 bases at a time. So which of the following codes for the amino acid leucine? So we're going to highlight those. So remember, if we wanted to read UUA, we would start from the center. U, we'd find the U and we'd work outwards. So we're now focusing on only this quadrant of our codon wheel. So U is the first letter, U is also the second letter, so we go out to this outer piece here, and then A is our last one. So we look here, A, I need to make this bigger, guys. UUA, that would be leucine, so we would go ahead and highlight that. So that's going to make leucine. Let's look at UUG. UUG will also bring in lysine. CUU, so start from the center and go out. C is the first letter, U is the second. And what did we say? CUU also brings leucine. UAA, UAA, that's a stop codon, so that would not bring in anything. And CUC, that also brings in leucine. All right, so which amino acid sequence is coded for by this RNA sequence? So we're doing the same thing now, we're just listing the amino acids. So UUU, U is our first letter, U is our second letter, and U is our last letter. That's phenylalanine. I'm going to abbreviate PHE. Our second one is GCA. GCA is alanine. AAG, AAG, that brings in lysine. And then we should be able to also go backwards. So there's going to be several possibilities that could work for these. So now we're going to look for the amino acid on the outside and just list a possible sequence that would work. So for PHE, that's phenylalanine. So let's find phenylalanine on here. We've been using this one on the top, phenylalanine. Our sequence could be UUC. What about serine? SER is serine. So let's find serine on the outside. Scanning, scanning. Oh, there's serine right here. And there may be other spots for it as well. UCA is a possibility. UCU, UCC, UCG, many different possibilities for serine, because that last letter, there's several options. So UCG. So that's how you would tackle those. I'm leaving these blank. You guys can fill in the rest. Basically, how do we tell DNA from RNA? So DNA is going to use deoxyribose. RNA is going to use ribose as the sugar. DNA has thymine. RNA has uracil. DNA has 2 strands. RNA is single-stranded. During DNA replication, you have 2 strands that are copied. So your 2 strands split and they each make a copy. So A goes to T, just remember base pairing, and C goes to G. In a strand of DNA, if there's 15 thymines on one strand, remember it's a double-stranded molecule, there must also be 15, just looking at our time, there must also be, if A and T go together, there must also be 15 adenines on the other side. The base pair. Variations occur at 13. So 13 is the base pairs. That's your base pairs. That's where we have variations. And I think that's it. So basically, we already looked at the nucleotide. So 12 is pointing to the circle. That's the sugar molecule on the sugar phosphate backbone. 11 is pointing to, nope, 12 is the phosphate. My bad. Phosphate. We just diagrammed our nucleotide here. Yep, so phosphates are circle. So that would mean 11 is the hexagon shape. That's your sugar. 10 is going to be a nitrogen base. And 2 nitrogen bases together for 13 makes a base pair. 9, here's another example of a nucleotide. You have a nitrogen base, a sugar, and a phosphate. So those 3 parts make up your nucleotide. Oops. And 8 is looking at the bonds holding those together. Those are hydrogen bonds. All right, hopefully you guys will hop onto tutorial or after school today on Zoom. Feel free to email me in the meantime.